This is Tyler with Crosscutting Concepts, and this video will be walking you through the ink chromatography procedure for the Mystery of Lyle and Louise Question Documents Kit. This lab experiment will test the ink composition of samples from 2 receipts to help establish their reliability. Begin by opening the development chamber. Fill the dropper with solvent and squirt its contents into the jar. And repeat this until 5 droppers full of solvent have been dispensed. Then close the development chamber and lightly shake or rotate to saturate the air inside. Using a ruler as a guide, very gently draw a horizontal line across the bottom of the TLC plate with a pencil, roughly 1 centimeter from the bottom of the plate. Be careful not to damage the plate surface. Then place 5 small dots with a pencil, spread evenly along this line. Label the dots A through E. On the data collection sheet, record the receipt number for the document to be tested. Fill in the receipt value and the person who signed the receipt. Then record the value of each digit on the data collection sheet, with 1 number per lane. Lane E will be reserved for the signature. Prepare a glass slide by placing it on a blank piece of paper. Then place the labels A through E beneath the slide. Carefully cut each of the 4 digits to be tested from the evidence receipt. Eliminate excess paper and avoid getting ink from more than 1 digit per cutout. Place the 4 cutout samples in order on the glass slide, by their corresponding labels. Then select and remove a small, ink-dense section of the signature for the final sample. Try to minimize damage to the signature to preserve the evidence. Each lab group will need to collect at least 5 microcaps from the microcap vial. Then place 1 drop of solvent on sample A. Draw a sample of ink into the microcap by gently touching it to the wet digit. Once the microcap has drawn up sufficient liquid, transfer the ink sample to the TLC plate by holding the microcap to the dot with the matching label. Repeat this process for each sample, being sure to avoid cross-contamination. When all samples have been transferred to the TLC plate, open the solvent jar and gently place the TLC plate within, with the sample end down. Do not allow the ink samples to directly contact the solvent. Lean the plate against the jar and close the container. The solvent should immediately begin carrying the ink upward while separating it into its component compounds. Allow the TLC plate to sit for 10 minutes, or until the solvent has saturated half the plate. While waiting, students may wish to work on the handwriting analysis lab. After 10 minutes, remove the plate from the chamber and place it on a blank piece of paper. Gently draw a line with a pencil to record how far up the plate the solvent reached. Then, allow the plate to dry. While waiting, repeat the preceding procedures with the other document in question. Once dry, perform the following steps for each receipt. Place a mark at the bottom of each ink component band. For each sample on the TLC plate, try to recognize the bands formed by each ink component. If a sample did not have enough ink, it may not be possible to read that sample. Rerun the experiment if you encounter this problem, being careful to collect enough ink. Using a ruler, record the distance between the sample starting line and the final distance for the solvent. In this example, the distance was 3.8 centimeters. This measurement should be recorded on the data collection sheet as total distance traveled by solvent, or D sub S. Measure and record the distance to each mark for each ink sample. You will also record the color of each ink component measured. The data collection sheet should appear similar to this after measurements are taken. Note that the first component in each lane was purple and had a short distance, and the last component was yellow and had a longer distance. Not all inks in our sample had 3 color components, so those areas were left blank. The retention factor for each component must then be calculated. Do so by dividing the distance the component traveled by the distance traveled by the solvent. Our first value will be 0.9 centimeters divided by 3.8 centimeters, for a 0.23 retention factor. Performing all calculations completes the worksheet for that receipt.